Well, good afternoon, everyone. My sincerest apologies that I'm not able to be there today. Iowa Swine Day is one of my favorite events of the year, and I'm excited about this program that's part of it as well. But USDA AFRI has their project directors meeting at the same time in Calgary, so I'm not able to be there. But I really appreciate the opportunity to US Soy to be able to present some of our work on soybean meal functional compounds and some of the data that we presented in one of our popular press articles about this topic. And so my goal for this presentation today is to help tease together some of the perspectives around soy functional compounds and human nutrition with some of the observations that we observed in animal science. Talk a little bit about their potential roles in the physiology of the animal, and then also discuss some of the limitations and future research areas to further investigate this aspect of soybean meal. So as nutritionists, we know that soybean meal is a superb amino acid source. It has a complementary amino acid profile to corn. It has a high lysine content, and we know that it is the main provider of amino acid in many commercial swine diets. However, in recent years, there's been more research that showed that when we displace soybean meal in the diet, even though we maintain non-essential essential amino acids, that there appears to be a decrease in growth and feed efficiency. And some of this has been related potentially to the functional aspects of soybean meal. Are there aspects of the plant that may be supporting the physiology of the animal beyond growth? And those may be towards helping mitigate viral load and certain disease stresses, or help mitigate inflammatory pressures and other stressors. Additionally, there's been more work that came out of Ryan Dilder's lab and Brooke Smith's dissertation that really studied isoflavones and what happens when we supplement isoflavones in grow-finished pig commercial production, as well as some of their mechanisms, and have shown that in these flavonoids, when supplemented to pigs, can reduce finishing pig mortality and help mitigate some of the growth response that's reduced with a PRRS challenge. Additionally, work that Kate Rush published out of the Hanor system has shown that pigs that are fed an increasing amount of soybean meal tend to have fewer cull pigs in those pens, fewer pigs that are pulled, and less medication treatments during the flu challenge. And so one of the questions we are asking are some of these functional compounds that are found in soybean meal that are not nutrients, but maybe supporting the physiology of the animal or mitigating disease, the infectaries of disease, or helping to support the immune system, partially responsible for some of these responses, not only relative to growth and to mortality, but also to energy. We think back to what Aaron Gaines was saying in his presentation before mine. We talked about the net energy of soybean meal. When we think about net energy in general, there's different components to net energy for growth, net energy for lipid deposition, net energy for protein, but we also have the net energy portion that goes to maintenance. And we can think about maintenance energy requirements are the amount of energy that's needed for basal metabolic functions to maintain body weight or to maintain homeostasis. We can easily estimate maintenance energy through a calculation, although the true maintenance energy requirement of an animal is never known, but is extrapolated from fasting heat production. When we think about maintenance energy and estimating it from metabolic body weight, we know that there's an increase in maintenance energy demand during different stressors. And so in the graph I have here on the left shows the maintenance energy calculation for a 30 kilogram pig based off of NRC 2012 equation. Oops. Over here on the right, I have a study by Van Meter et al. That showed that high health pigs at the same weight have very similar maintenance energy requirements to our prediction equations that were extrapolated from fasting heat production. If we take those same pigs and put them in a sanitary challenge, their maintenance energy requirements increase by about 10%. Similarly, Nicole Huntley showed in her PhD dissertation work that if we take about the same size of pigs at 30 kilograms and inject them with lipopolysaccharide, which is an immune system stimulator, that the maintenance energy requirement of that pig increases by about 40%. And the reason I show this is that when we think about net energy values or net energy in general, is that net energy is not only is a dynamic measure. And so assuming a constant maintenance energy requirement is oversimplification of reality. And so when we're estimating net energy caloric efficiency, or when we're using caloric efficiency, we have to consider the maintenance energy requirements of those animals are likely different to that in academic settings. And so one of the hypotheses that we have is that our functional compounds, or compounds that may help mitigate inflammation or mitigate the amount of energy that needs to go to the immune system or other various physiological processes, partly contributing to maybe the increased energy that we observe in soybean meal. And so we know that these compounds have anti-inflammatory, antimicrobial effects, both systemically within the pig, but also within the GI tract. So what is a functional compound? We can heard them referred to in many different vectors in the literature, in other publications as either nutraceuticals, bioactive compounds, non-nutritive compounds. They take on a variety of different names. More recently, I've been heard them referred to as bioactive compounds, but in strictly scientific sense, bioactive is an alternative term for biologically active. And so any organic molecule that has biological activity is considered bioactive. An amino acid is bioactive. A carbohydrate is bioactive because it is metabolized. What separates these compounds from nutrients? Well, nutrients have a requirement. These functional compounds don't have a requirement, but they can be a component with a selective effect or one or various functions of the organism. And there's no direct established scientific definition for a functional compound, but Dr. Arneson, a human nutritionist, proposed one in 2016 that I think is really encompassing when we think about the functional aspects of soybean meal, and that a functional compound is a component with a selective effect or one or various functions of the organism that can be justified as physiological or healthy. He also gives three characterizations of a functional compound that it must have to be considered one as a functional food in human nutrition. Has to be derived from a naturally occurring ingredient, has to be consumed as a part of a daily diet, and it has to be involved in regulating a specific physiological process. So if we think about soybean meal in the context of swine nutrition, it's derived from a naturally occurring ingredient. It is consumed as part of the daily diet in a pig, and it can, in these functional compounds, and from the data I'm going to show you, is involved in regulating specific physiological process, depending on which compound we're referring to. One of the other aspects of functional compounds that I want us to be aware of and keep in the back of our mind is that many of the functional aspects of these compounds that are found in soybean meal and in other ingredients as well can be considered anti-nutritive in human or anti-nutritive in animal nutrition, but are positive in human nutrition. For example, we know that protease inhibitors reduce nutrient digestibility and amino acid digestibility and can help and have a huge influence on growth performance. But in the human nutrition space, they're thought as beneficial as that they have known anti-carcinogenic effects. And so when we think about the terms of functional compounds and we look at the vast amount of science in this space, you have the context is really important because for us, we may be looking at a 6 of these particular compounds, and human nutrition and human medicine is looking at a 9. And so when we think about these functional compounds, context is important. And I also want you to think outside of the box. There is no box relative to classical nutrition because these are not necessarily having the same effect as we think of in nutrition, even though it's part of the diet, but they're really physiological mediators when they're bioavailable. So what are the different functional compounds in soybean meal? So if we take, there are over 40 different functional compounds that can be found in soybean meal or in soybeans themselves. And then if we look at those that are present in soybean meal and consistent in high enough concentrations, we can divide those into 4 different categories. And so we have categorized them in polyphenols and terpenoids, which would include isoflavones, saponins, and polyphenols. We have prebiotic oligosaccharides and fiber, functional lipids, which would include phospholipids, lecithin, some of the addbacks as part of soybean meal processing, and fatty acids, and a little bit of phytosterol, such as beta-sigmasterol, and then also bioactive peptides, which are peptides that are released from the protein subunit itself as part of partial unintact digestion. And so if we group these 4 compounds together, we think about all the functional compounds that could be in soybean meal, they represent about 70% of those compounds. And today I'm going to focus on the first 3 categories, as those appear to have the most impact on the physiology of the pig in the context of animal nutrition. So we put together an article describing some of these compounds and their potential role, but only in the animal, but in the plant as well. And so the first category of compounds that I'm going to talk about are polyphenols and terpenoids. These are going to include things like phenolic acids, isoflavones, and saponins or terpenoids. These have a variety of different functional roles, but what's interesting about these compounds is that they all derive from one singular pathway within the plant itself. So there's this pathway within soybeans as they're growing called the phenylpropanoid pathway, and it's really critical to plant biology. It plays a vital role in plant development by acting essential components of cell walls. It protects those plants against high light situations and UV radiations. It helps during abiotic stress and when there are plant pathogens. It also helps to give some of the pigmentation to soybean as well. And so these phenolic acids and isoflavones are incorporated into the cell wall of the plant in response to biotic stress. And so there's an increase in this particular pathway depending on the environment of which the plant is in. And so we have to think about that the inhibition and activation of this pathway is heavily influenced not only by growing conditions, but cultivars as well. Well, why is that important in terms of the context of human nutrition? Well, the concentration of these compounds can be very diverse and highly variable from soybean meal source to soybean meal source. Additionally, a lot of these phenolic acids are precursors to other flavonoids within this pathway. And so depending on the state of maturity of that plant, as well as the stress state of that plant, will help dictate how much of these compounds are present within the plant cell wall, and then finds its way into soybean meal. So we think about the first category of those polyphenols. The first one would be phenolic acids. So there's quite a variety of phenolic acids that are found in soybeans. Benzoic acid, cameric acid, ferulic acid are probably the main ones, with a little bit of ginsetic acid and as well as vanillic acid. These are more concentrated in soybean meal than they are in the intact soybeans themselves, because they are largely associated with the protein content of the soybean. And so soybean meal is 37 to about 60% more concentrated in these than the whole soybean. But their bioavailability is quite variable. Animal and nutrition and human nutrition literature would say that they're vastly released in the large intestine, and they have variability that ranges between 15 and 40%. Physiologically, these polyphenols are known as potent antioxidants in that in oxidative stress states, or they act as an antioxidant within the body, that they help to scavenge free radicals and help to neutralize them and act as antioxidants. And so that is their major physiological role within the animal. Isoflavones are derived from many of those phenolic acids in that pathway that I was discussing. Isoflavones are a class of flavonoids with antioxidant, anti-inflammatory, and antiviral properties that have been shown in a variety of different human nutrition and human medicine and cell culture realms, and here more recently in some of the animal nutrition work. There's two major isoflavones found in soybean meal, genistein and diazidine. They're also more concentrated in soybean meal than they are in the intact soybean. And we know that these can have a role in physiology, as they can act as phytoestrogens, which is largely their study in the human nutrition realm, and that they may mimic estrogenic activity and contribute to plant defenses. However, one thing we have to keep in mind about these isoflavones is bioavailability. When they are intact in the plant, like in this graph on the right, they're bound to a carbohydrate subunit. And so in order to be bioaccessible to the animal, they have to be digested, or a beta glycosides has to release them and put them in their aglycone or active form. And so the active forms have an E in their name. And so genistein is the active form of that genistein isoflavone. And so they have to be in their active form to be bioavailable. And so that's one of the limitations in understanding isoflavones in intact soybean meal, is we don't have a good understanding of their bioaccessibility. How bioavailable are they to the animal? We do know that the large proportion of them that are released from their carbohydrate subunit are actually metabolized in the small intestine. And so there's not in the human sciences, there's not as much that makes it systemically. So they're largely focused within the gastrointestinal tract locally, somewhat systemically. They may be absorbed in the small intestine and go through the basolateral side, but the efficacy and flux of that is largely unknown. The interesting thing about these isoflavones as well is that they can be fermented or metabolized by microbiota into other unique metabolites, such as equal, which are really potent antioxidants and antimicrobials within the gastrointestinal tract, that are known to have known effects on streptococcus and Clostridium perfringens. And so there are also their metabolites from the microbiome can have an influence on that gastrointestinal lumen and in the health of the animal. More recently, though, isoflavones have been shown to have more antiviral activity. So there's a really great review article. It's a little bit dated from 2009 that talks through about the different potential mechanisms of how isoflavones, particularly genistein, can inhibit viral entry and replication. And this has been largely associated with the functional aspects of soybean meal in PRRS infected pigs, is that it may help to inhibit viral entry, replication, largely through the protein tyrosine kinase pathway. And shows that it may be able to mitigate that and help prevent viral entry. However, we still have some unknowns relative to this because a lot of the really potent data that we've showed relative to PRRS are fed, those pigs are fed isolated isoflavones. And so we have to consider the bioavailability and bioaccessibility of that particular compound and whether or not it can influence the viral activity within the animal that maybe succumb to a viral challenge. Outside of their antiviral activity, we've seen some really interesting data in white blood cell counts in nursery pigs. So one of my master's students back when I was at Texas Tech University, Brooke Bowen, her master's thesis was really looking at some of these compounds in the weaned pig and during that critical inflammatory transition state. And so we fed 6 different diets. On the far left, we have is a soybean meal control diet. So this is pushing quite a bit of soybean meal, about 27% in a phase 2 nursery diet. On the right, we have a control diet that has soy protein isolate in it. And then we took that control diet with soy protein isolate and we added back in these 4 categories of functional compounds to closely meet the amount of functional compounds that were being fed by the intact soybean meal. So we have isoflavones added back here to help meet about this amount of isoflavones that would be found in intact soybean meal, lipids, fiber, and as well as lunasin, which is one of those bioactive peptides. Interestingly, when we did CBC counts of these nursery pigs, that in both the intact soybean meal diet and in the diet supplemented with isoflavone, we see a 33% reduction in neutrophils observed. And this is a mechanism that is also been shown in cell culture work and in mice models that genistein can have a systemic effect on neutrophil suppression by mediating 2 known pathways, MAPK and NFK beta, relative to the phagocytosis of those particular cells. Well, why does that matter? Well, we know that circulating neutrophils are part of the first line of defense of the immune system of the animal. And so potentially, are we reducing their overall concentration or are we helping mediate them to go into areas and sites of where there's inflammation? And so it was one of the interesting responses that we observed from this study. We also observed that these neutrophils act a little bit differently and that they have different reactive oxygen species bursts compared to that soy protein isolate control. So not only is the concentration changing, but the circulating neutrophils that are there are acting a little bit differently when they're exposed to both intact soybean meal and soy isoflavones. Additionally, when we look in the gut of these pigs, same treatments here, just colored bars now, and in the same order, is that the total antioxidant capacity of the ileum tissue is considerably higher in pigs fed both intact soybean meal and those that are fed in soy isoflavones. We know that isoflavones can combat oxidation by neutralizing free radicals and inhibiting singlet oxidation without producing additional radicals. This is really important because that antioxidant can recycle. So it's more potent than other antioxidants because it is able to mitigate multiple reactive oxygen species because it's able to recycle itself as an antioxidant. And so this observation has also been observed in other studies with nursery pigs that were fed isolated isoflavones. So when we think about isoflavones and their potential role in the diet in terms of the animal, they're largely antiviral, antioxidant, anti-inflammatory in terms of their potential mechanisms there. But one of the key things we have to think about is bioaccessibility and bioavailability. We move on to that second category of compounds, really talking about dietary fiber and oligosaccharides. So dietary fiber in soybean meal is largely cellulose, ramilogalactans, or pectin-based substrates, as well as very high in non-digestible oligosaccharides. stachys, raffinose, and verbicose. And we know from these in animal nutrition as potentially having adverse effects in the young pig, increasing scouring effects, and that they're highly fermentable. And that is certainly true. When we think about them in a functional context, it doesn't necessarily take away that they have anti-nutritive effects in young pigs. That's not the case. But when we think about that mid-grower pig, are they acting particularly in a different role? So we know soybean meal has a decent amount of fiber. It's about 16% total fiber when we count in those oligosaccharides. And we know that those lactose oligosaccharides in the human nutrition realm have a prebiotic effect. Well, what is a prebiotic? Prebiotic is any substance that is selectively utilized by host microorganisms and confers a health benefit. So what are some of the prebiotic effects of galactose oligosaccharides? There's a really nice review paper that is in journal Frontiers in Animal Nutrition that talks about or Frontiers of Nutrition that talks about the galactose oligosaccharide effects of soy verbicose, galactose oligosaccharides. And so these can be fermented by microorganisms that are known to confer a health benefit. Those would include lactobacillus and bifidobacterium. Having an increased enrichment of those communities helps to deter other pathogenic bacteria. And we know that those two communities work cooperatively together to produce short-chain fatty acids, particularly butyrate. And if you've paid attention to the short-chain fatty acid data, we know that butyrate is a high potent modulator of a variety of different cell signaling pathways within the GI tract, particularly those that are associated with increasing tight junction proteins, immune regulation, regulating the proliferation of immune cells, mucus secretions, as well as butyrate can be utilized as an energy source. And so when we think about the fermentation of these, if they are smaller oligosaccharides, are more highly fermentable. And so those shorter chain galactose oligosaccharides may be conferring a health benefit in that grower pig while still pertaining their properties of being anti-nutritive in that young pig. So remember, it's all about context. And so in one of our studies, we observed that we fed those soy fiber concentration back to the pig and as well as intact soybean meal, that we see an increase in the molar proportions of short chain fatty acids within the ileum and in the colon of those pigs. But because these were weaned pigs, we still saw the anti-nutritive effects that are associated with these. And so really in that grower pig to early finisher pig, when we're feeding a lot of intact soybean meal, could those galactose oligosaccharides be playing a role in terms of as a prebiotic like we see in human nutrition? The last category of compounds I'd like to talk about is one that's not as prevalent in soybean meal, and that would be functional lipids. And so the main functional lipids that are found in soybean meal are phytosterols, camphosterol, beta-sigmasterol, and sigmasterol are the main three, as well as there's a few isotypes of those, as well as phospholipids. So phosphatidylcholine and phosphatidylinositol can also be found in soybean meal, and particularly in soybean meals. Plants that are adjacent to oil manufacturers that may have addbacks added back onto them tend to have a higher concentration of phospholipids. So what do phospholipids and phytosterols play a role in the animal? Well, we know in human nutrition that phytosterols can exhibit anti-inflammatory and antioxidant properties like we've discussed previously, but we also know that phospholipids can serve as an energy source and are required for structural stability of cell membranes. They help with the reserves for choline within the animal and are involved in secondary messenger systems of systems of signal transduction. Some other potential benefits of soy lipids is that they have ability to impact red blood cells. And so red blood cells that hold in more chylomicrons relative to soy lipids have increased nutrient flux and oxidation, and particularly that's been associated in the human nutrition realm to lecithin. And in our study, we observed a similar response in that pigs that were fed the red blood cell count of pigs that were fed isolated functional lipids from soybean meal had increased hemoglobin and hematocrit. And so this can be a largely result of the soy lipids increasing fatty acid content within blood and then being able to increase movement and permeability of red blood cell counts. Take-home message around functional lipids is it's probably one of the lower impacts it has in the physiology of the animal. The diversity of the concentration of those compounds in soybean meal are quite variable depending on how much oil is being extracted and how much of that is left in the solvent fraction. And so we place less pertinence on this category of functional compounds, but they are still present in many soybean meal sources. So thinking about my take-home messages of what I'd like for you to get from this presentation today is that soybean meal may play a role in improving growth viability and reducing the need for medical interventions. And this may be attenuated by some of these functional compounds, particularly as it relates to viral replication in PRRS. However, we have to think about that a lot of this is not necessarily cause and effect. It's a lot of association-based study and understanding at this point. And so really we need further work to help completely delineate the potential mechanism of these compounds in the terms of intact soybean meal and under commercial conditions where we have those higher maintenance energy costs. When we think about the energetic value of soybean meal and we're trying to understand its potential role, and I would say this applies to many ingredients as we think about their role in the diet, is that altering maintenance energy is also an impact that an ingredient or an additive can have. We know that an increased pathogen load and changes in immune cell metabolism and inflammation all require ATP. And so that's going to have a demand for energy in terms as it relates to the net energy value of an ingredient. And then lastly, the bioavailability and metabolism of these functional compounds right now is poorly understood. And so we need further research to help understand when we feed intact soybean meal, how much of these functional compounds are able to actually have an effect on the animal and what is their bioavailability in that sense. And so I won't be able to take any questions because I'm not there in person today, but I look forward to your questions in the question and answer portion where I will be available later today. And none of the research I presented today relative to our group could be done without this team that you see here. Thank you so much.